let me introduce today's speaker. It's my great pleasure to introduce Dr. Steve Lacroix. Steve obtained his Ph.D. from Laval University in Canada in 1998 and went on to do his postdoc work in UCSD and then in McGill University. He's currently a professor in Laval and the director of a neuroscience research center there. Steve has been a very successful neuroscientist and a neuroimmunologist. He has garnered many awards for his research work and published numerous papers in top-ranked journals. Because both of us love as you can see from the title, we both love to study the IL-1 system, and we become friends and collaborators. So I have always admired Steve's wonderful insight and his very systematic approach to his research. And I myself have learned a great deal from his work. And I'm sure the students here will learn a lot from this lecture as well. So just a reminder, if you have questions, please raise them in the Q&A box. And without further ado, Steve.
Thank you so much, Ning. Everybody can hear me well?
Yes.
Awesome. Outstanding. So thank you so much to Ning, Quan for this wonderful invitation. And also thank you to Dr. Randy Blakely for taking the time to meet with me today. It's really greatly an honor to be able to speak in front of a nice crowd from the Florida Atlantic University. I'm happy to be here today. I'm going to share my screen. I really wanted to thank once again Ning, Quan, Jennifer for the help, and also Dr. Blakely. So I'm going to start without further ado, just to make sure that I'm on time. Today, I will talk about the involvement of the Il-1 Cytokine System in CNS pathologies. This slide is just to declare that I have nothing to declare. And I'm going to focus today in my talk on a disease called multiple sclerosis that you quite know, I'm pretty sure, quite well. So multiple sclerosis is a chronic inflammatory disease of the CNS. Many believe that it's an autoimmune disease of the CNS. It's mainly characterized by the attack of the myelin and also the myelin by the myeloid cells, the T and B lymphocytes. And although the etiology of the disease is quite obscure, there's many factors that have been identified. Recently, a very nice study in Science has shown that the Epstein-Barr virus might be a risk factor. And we all know that vitamin D, smoking, and also biologist sex is involved. But this is just to briefly introduce you to the disease. When I was used to travel 15 years ago, everybody was talking about MS being a brain-specific disease. But I just came back from a meeting over the last few days where it's more and more recognized that, well, the spinal cord is quite affected by the disease as well. So there's many plaques, many infiltrates of immune cells that are in the spinal cord per se. And I'm interested about the spinal cord since many, many years. In fact, I've always been interested about spinal cord pathologies overall. And why am I interested about spinal cord? It's because it's a relay system that is quite important because it connects the periphery with the brain. Everything that goes through the brain and needs to be connected with the periphery has to pass by the spinal cord. So the spinal cord is important when it comes to inflammation because it regulates basically the immune response in any type of organs that exist, starting with the bone and the bone marrow, the spleen, the lymph nodes. So that's why the spinal cord is so important. Coming back to the main subject of the talk, I think you can see my cursor here. I'm going to talk mainly about the IL-1 cytokines, mainly about two of these cytokines, in fact, IL-1 beta and IL-1 alpha. Those two cytokines interact via a common receptor, which is the IL-1 receptor type 1, which I'm going to call during my talk as IL-1 receptor type 1, IL-101. The two cytokines are different in the sense that IL-1 alpha is constitutively expressed within the cells. When we talked about the CNS, it's constitutively expressed into microglia. We know that it's in the nucleus of these cells, it seems that it has a role in the regulation of gene transcription into the microglia. But when there's damage to these cells and these cells undergo necrosis, IL-1 alpha is being released and can orchestrate inflammation because IL-1 alpha and IL-1 beta are kind of the chef d'orchestre of the inflammatory response overall. But today I will mainly focus about IL-1 beta because IL-1 beta has mainly been involved in mechanism of infection, for example, and also in disease related to autoimmunity, which is one of my main interests. So IL-1 alpha mainly involved in sterile inflammation, the kind of inflammation that will develop after traumatic brain injury, spinal cord injury, ischemic stroke, whereas IL-1 beta is mainly involved in autoimmunity and also in infectious disease. As I pointed out, both of these cytokines signal through IL-1 receptor type 1. IL-1R1, once it's activated, will actually form an heterodimer with IL-1 receptor accessory protein. Eventually, this will recruit, mediate, and IRAC and signal through a quite complex signaling pathways involving MAP kinase, like P38, for example, Janus kinase, and also ERK, and also NF-kappa B. The end results of this signaling pathway is really activation of transcription of genes involved in inflammation. And among these genes are cell adhesion molecules, proteases, cytokines, and chemokines, and so on. So really, IL-1, the IL-1 system is a key cytokine involved in inflammation, especially in chronic inflammation that you will observe in autoimmune disease, for example. The IL-1 beta is quite complex because it requires two different signals to be to become bioactive. I told you that IL-1 alpha is constantly produced as a proform, and the proform of IL-1 alpha is bioactive. But in the case of IL-1 beta, it's different because it's not constitutively present within the cells. You need basically to have synthesis of the transcript that eventually lead to the production of the proform of IL-1 beta. And this proform needs to be cleaved to become bioactive. There's two different signals that will lead to that, and I will come back in a few slides about these two different signals that will eventually lead to the transcription, to the formation of the proform, and eventually the cleavage into a bioactive form of IL-1 beta. So what is the evidence that IL-1 beta system has a role to play in multiple sclerosis? Well, IL-1 beta is present in the brain lesions. It's present also in the CSF of individuals with multiple sclerosis. And the amount of IL-1 beta correlates quite well with the cortical lesion load in these patients. There's many examples as well of families that are characterized with a high ratio of IL-1 beta over the IL-1 receptor antagonist, which is a natural antagonist produced in the human body. So those that have a higher ratio of IL-1 beta over IL-1 receptor antagonists are more predisposed to develop autoimmune disease, including multiple sclerosis. And finally, but not least, peripheral blood mononuclear cells of MS patient, well, they do express significantly higher levels of hemerin and also protein for IL-1 beta, caspase-1, and some of the NOD-like receptor. And I'll come back to the NOD-like receptor a little bit later on. Finally, as July 2019, there has been an ongoing clinical trial led by the National Institute of Neurological Disorders and Stroke in the US for the treatment of chronically inflamed white matter lesion in MS using anakinra, which is a formulation of the natural antagonist of IL-1 receptor type 1. So in my lab, we are interested in many different diseases. As I pointed out, autoimmune disease are quite an interest of us since a long time, in particular multiple sclerosis. And for studying multiple sclerosis, we use basically a mouse model, which is called experimental autoimmune encephalomyelitis. This mouse model constitutes in the injection of the MOG, myelin oligodendrocytes, glycoprotein 3555, it's a peptide. And once you inject this peptide and you infuse complete Freud adjuvant, followed by a couple of injections of pertussis toxin, you end up with a disease that shares many similarities with multiple sclerosis. And we typically look at the clinical score for animals to be able to determine whether the animal is sick or not. And on this scale of 5 points, 0 mean a completely normal animal, whereas 5 means completely paralyzed and even dead in some of these animals. As this crowd is aware, there's many different forms of multiple sclerosis. In my lab, we're studying the chronic EAE mouse model, which recapitulates some of the form that you find in many individuals that suffer from the disease. Normally, typically, we use this mouse model because it's in the B6 mice, and B6 mice happens to be the background of many of the transgenic mouse lines that I will show you in the next few slides. Coming back to the importance of the IL-1 system, why we became interested about IL-1 itself, I think that this slide summarized quite well why we became interested. We did some experiment early on with some of these mouse models that are deficient in IL-1 alpha that you see in panel B, IL-1 beta, or the receptor in panel A. And what we found is that when you immunize these mice to develop the disease that shares similarities with multiple sclerosis to develop EAE, it turned out that the IL-1 alpha knockout mice have basically no effect, no change when you compare the IL-1 knockout mice with the knockouts compared to the wild type. However, when you look at IL-1 deficient, IL-1 beta deficient mice, these animals barely develop the disease. In fact, about 30% of the animals immunized will actually end up with the disease, and it's always going to be a very slight disease. The maximal score that we typically see in the IL-1 beta knockout will be about 1 on this grade scale of 0 to 5, which means that paralysis is only of the tail in these animals. The IL-1 receptor knockout mice recapitulate many of the effects that you're seeing in the IL-1 beta, meaning that it's a very minimal disease that you're seeing in these animals, and typically the disease is not very severe. In panel D to F, you basically see some work that was done with bone marrow chimeras. So what we do with these animals is that we irradiate the animals. It's a lethal irradiation. And then you transplant these animals with bone marrow, because if you don't, they will eventually die after a couple of weeks. And what you can do with these chimeras is that depending on the donor and also the receiving hand of the animal, you can actually generate chimeric mice that will be wild type into wild type into knockout, or vice versa, and knockout into knockout. And this is really important to understand what is the compartment that is important for the production of IL-1 beta, or for the cells and identifying the cells that are responsive to IL-1 beta. As you can see, once again, if you develop chimeras with the IL-1 alpha, there's no effect, no change between wild type and knockouts. But if you look carefully at panel F, if you irradiate the mice, transplant them, the one that recapitulate the effect that you're seeing in the knockout is the animal that will have actually a bone marrow that will be knockout in IL-1 beta, and that will be given to a recipient mouse line that will be a wild type. This suggests that the immune system is really the compartment where the IL-1 beta protein is being produced. It does suggest that. On contrary, if you look at panel D, if you look at the different chimeras, those that recapitulate the effect of the knockout into knockout are the wild type into knockout mouse line, suggesting that the radio-resistant cells are the ones that are responsive to IL-1 beta. So it does suggest that those could be CNS resident cells, such as neurons, endothelial cells, or some kind of glial cells, or endothelial cells per se. So then what we became really interested is to be able to identify some of these cells. So the research question we are asking is, what are the effects of IL-1 beta in the pathogenesis of multiple sclerosis and the comorbidities that are associated with the disease? In that case, I'm talking about the pain response, because pain has been really a big problem in the individuals that are suffering from multiple sclerosis, unfortunately. Her hypothesis is that it's the migration of the myeloid cells, of the immune cells, across the inflamed blood-brain barrier, blood-spinal cord barrier that will trigger an IL-1 beta, IL-1 receptor type 1 signaling pathways that eventually will lead to development of the disease and chronic pain. So the two main objectives of our work of this presentation will be to identify the cells that produce IL-1 beta and the receptor in the context of multiple sclerosis and in the animal model of EAE, and eventually assess what will be the effect of this activation of the signaling pathway IL-1 beta, IL-1 receptor type 1 in the development of the disease per se, and also in pain. But first, let's start with the cells that produce IL-1 beta. I'm not going to let you linger too long. Actually, we tested many antibodies over the years, and very few of them are quite specific. This one is. This is the antibody that has been produced by R&D System. It's a commercial source. It's a company. We've tested these antibodies in many ways in the knockouts. We have also pretreated the tissue with anakinra and block basically, which will block the interaction between the antibody and its binding site. And the signal was completely gone in these two conditions. So we have validated this antibody in many ways. And when you use the anti-IL-1 receptor type 1 antibody and you stain brain, you stain spinal cord tissue, the first thing that you're seeing is really intense signal in the blood vessels. You see here in red, this is IL-1 receptor type 1 immunostaining. And you see in panel A and B in green, the staining with a specific marker for endothelial cells of the blood vessels. As you can appreciate, it's not all type of blood vessels that do express IL-1 receptor type 1, but when the signal is intense, the signal is quite intense though, but not in all type of blood vessels. This is the basement membrane, and you're seeing in white here with the merged image on the right side. If you stain and you try to colocalize with alpha smooth muscle actin, which is a marker of pericytes and fibroblasts associated mainly with arteries and arterioles, what you find is that most of the IL-1 receptor signal does not colocalize, suggesting really that IL-1 receptor type 1 is mainly expressed in blood vessels of the venous system. But what is the time course of expression of the IL-1 beta protein? So to be able to understand when exactly IL-1 beta is being produced, even though some good antibodies exist that allows you to detect IL-1 beta, those antibodies do not allow you in immunostaining to discriminate the pole form from the cleave form of IL-1 beta. Most of these antibodies, if not all, actually detect both forms. So the only specific way to be able to determine with certainty that you are in presence of the mature bioactive form of IL-1 beta is to use immunoblotting and to be able to look at the molecular weight of the protein. So this is exactly what we have done. We have immunized animals, sacrificed some animal at 0, 1 day, 3 days, 7 days, 11 days, and 14 days post-EAE. You can see in panel 8 here the immunoblotting results. And the bottom line, the take-home message of this figure is really that the cleave form, bioactive form of IL-1 beta is being produced at the onset of disease and at the peak of disease. When you look at early time point, before actually day 10, where the animals don't show any sign of sickness, you don't detect IL-1 beta in the CNS. This is nicely correlated with at the transcript level when you look at the same time point, IL-1 beta and caspase-1. I'll come back to caspase-1 because this is the enzyme responsible for the maturation of the pole form of IL-1 beta to the mature bioactive form. Once again, it's nicely expressed mainly at the onset of disease and at the peak of disease. To be able to understand and look more specifically at the cell type capable of producing pro-IL-1 beta, we've turned to a collaboration with a colleague at University of Toledo. And this researcher was able to produce a reporter mouse line, basically expressing DS-RAD under the control of the IL-1 beta gene promoter. So he did develop this mouse line. He was kind enough to send the mouse line to us. We've immunized animals once again. We've sacrificed animals at different time points. And using the spinal cord of these animals that have been immunized, we were able to dissociate the cells from the brain, from the spinal cord. This is spinal cord tissue that you're seeing here. And to be able to examine basically the expression of the reporter DS-RAD in various cell types. As you can see in panel D, there's neutrophils, so some type of myeloid cells, myeloid immune cells, neutrophils, monocyte-derived macrophages, MDM, microglia, and T cells. And as you can appreciate, this is flow cytometry results. As you can appreciate, the main producer of the proform of IL-1 beta are really neutrophils and monocyte-derived macrophages. You barely detect any IL-1 beta in the microglia and the T cell population during EE. This is once again further demonstrated using the reporter mouse line I was telling you about. This is the DS-RAD negative population. You don't detect the transcript coding for IL-1 beta or caspase-1 barely in the cell population. However, when you go and look at the DS-RAD positive cell population, and more specifically at the monocyte-derived macrophages and neutrophils, those are the cells that heavily express the transcript for IL-1 beta and caspase-1. This is how it looks like when you immunostain the tissue from these animals. This is the reporter mouse line that has been immunized. So there's a lot of immune cells that are stained in the tissue. Most of the time in the disease, EAE or multiple sclerosis, they accumulate and they infiltrate next to the blood vessels or close by the meninges. You see a lot of infiltrates of these cells. And when you try to colocalize with markers of immune cell subset, this is a marker of neutrophils, LY6G. This is a marker of T cells, CD3, and this is a marker of macrophages and microglia, IL-1. You only see nice colocalization with LY6G and IL-1. It does not colocalize with the T cell marker. Once again, suggesting, and again, I mean, you see with the white staining, the basement membrane that has been stained with laminin. So you can appreciate that these infiltrate of immune cells are mainly associated with blood vessels. This is from where they are coming in the spinal cord in the brain. They infiltrate from the blood vessels. And you can see that it's a nice colocalization with the quantification on your right, nice colocalization with the neutrophil marker and also IL-1 marker. So it's roughly about 70% of these cells are neutrophils and 30% of these cells are macrophages. So we next decided to use intravitreal microscopy technology to be able to understand what exactly in time are these cells producing the IL-1 beta or the pro-IL-1 beta in that case. So we were able to use, once again, the reporter mouse line that I talked to you about before, the DS-RAD promoter, the IL-1 beta promoter that drives expression of DS-RAD, mainly neutrophils, as I pointed out, and also in monocyte-derived macrophages, because those are the cell types producing the pro-IL-1 beta. And we crossed this mouse line with the LISAM EGFP mouse line. This transgenic mouse line expressed basically GFP in green, specifically in cell of the granulomyelomonocytic lineage, meaning mainly, once again, neutrophils and monocyte-derived macrophages, if we're talking about the tissue. Here you will see in this video what we've done with the technology of intravitreal microscopy in the spinal cord. So we anesthetize the animal that have been immunized. We suspend the animal on the stereotax, and then we can image the spinal cord live. You will see the staining in green will be the staining that you will observe in perivascular macrophages. You will observe that also in monocytes and as well in neutrophils that travel in the blood. The blood will be stained with a Q dot. It will appear in blue. And the reporter mouse line for pro-IL-1 beta will be in red. I'm going to start the video. This is pre-onset. The animal are not sick at this time point. And as you can appreciate, there's a few perivascular macrophages in green. There's some cells neutrophils and monocytes that travel in the blood, but you don't see anything in red. There's none of these cells, basically. They don't express the DS-RAD protein before the onset of disease, at least not in the cerebral vasculature of the spinal cord. What is happening, though, when you look at a time point like onset plus 1 day? So at onset plus 1 day, the animals are starting to be sick. They already show some symptom of paralysis. When you look at that, the situation is completely different. I'm going to start the video here. Already, when you start imaging at this time point, you can already appreciate that in the CNS parenchyma, in the spinal cord parenchyma, there's some cells that are expressing pro-IL-1 beta. And those cells nicely colocalize. You see it on the right panel. They nicely colocalize once again with the reporter mouse line that allows you to look at neutrophils and monocyte-derived macrophages. I'm going to start the video again, and I want you to focus this time on the cells that travel in the blood vessels, because you will see some example of these cells being firmly adhere. There's one example here and another one here. So those cells that are firmly adhere to the lumen of the blood vessels do express as well the proform of IL-1 beta. So we did perform some quantification. This is an example that was done with postmortem tissue that was fixed and stained. As you can appreciate, pre-onset, there's not much going on in the DS-RAD panel. However, when you look at onset of disease, you see a lot of these cells in the spinal cord parenchyma. They colocalize once again with the marker of LISM positive neutrophils and monocyte-derived macrophages. And here I would like you to pay attention to the quantification that was done on the bottom right panel. You can see that the circulating cells, the rolling cells, they don't express DS-RAD protein, meaning that they don't express pro-IL-1 beta. However, when they start adhering, it seems that the process of production of the proform really starts. And this is really when the cells are reaching the CNS parenchyma that they do express a lot of the proform of the protein. So in a quite productive collaboration that we started with the laboratory of Alex Pratt, he's a neurologist, he's an expert in multiple sclerosis. We wanted to ask the question of whether it's possible or not that these cells, it's the migration of these immune cells through the blood spinal cord barrier, through the endothelium, basically, that is kind of the trigger of the production of IL-1 beta. And this is exactly what we tested here. We took out some GR1 positive cells from the bone marrow. Those cells are neutrophils mainly and monocytes. And we use in that case some of these cells coming from the DS-RAD reporter mouse line. When we place these cells into a Boyden chamber in which we had basically culture brain microvascular endothelial cells at the surface of the Boyden chamber, it's a transmigration assay. And you place these immune cells on top. What they will do naturally is that they will cross the endothelium, because that's what immune cells are doing in the context of stimulation. And when you look at DS-RAD expression in the upper compartment, you don't see a lot of DS-RAD expression. However, if you look at the cells that have transmigrated across the endothelium, it turns out that there's a lot of expression of DS-RAD, suggesting that this is really the transmigration of these cells into an inflamed tissue that could be the trigger of the production of the proform of IL-1 beta. So coming back to this slide, I told you that the production of IL-1 beta was quite complex. It involved two different signals. The first signal is the signal that normally would trigger the production of the transcript. It's been known over the years by many groups, including the group of Ning, Quan, back in his old days when he was working with Miles Zirconam, that some of these signals are actually the toll-like receptors. If you stimulate the toll-like receptors, you will activate the transcription of the transcript for IL-1 beta. And eventually, though, the second signal that is needed once you have transcription, production of the proform, is really the cleavage of the proform into the bioactive form. And it's been shown over the years that the The second signal could be, for example, nucleotides such as ATP, which is produced during infection, during cell damage, for example. So ATP seems to be quite important. And it's important because it does activate what is known as the inflammasome. The inflammasome is a multi-protein complex form of many different proteins that eventually will lead to the cleavage. So what it does is that it's composed of biosensors, which are the node-like receptors. There's many different types of node-like receptors. It's quite complicated. Each one of these node-like receptor will recognize a different microbe. Most of the time, they will recognize a pattern that will be replicated basically in many different kind of microbes. And this node-like receptor will link eventually the adapter protein called ASC. ASC, it's an adapter protein that is important because it will link on one side the inflammasome, the node-like receptors, and also will recruit procaspase 1. Procaspase 1 is important because it's the enzyme that needs to cleave eventually IL-1 beta to generate the mature form. So this is highlighted here. So for my talk, I want you to remember that ASC is a key adapter protein involved in the formation and activation of inflammasomes. So what happens during EAE in terms of the expression of these different inflammasome proteins? Well, I showed you before, IL-1 beta and caspase 1 are upregulated at onset of disease, in this case around day 10, and also at the peak of disease, which is typically between day 12 and day 14 following immunization. So both IL-1 beta and caspase 1 are highly produced. This is actually spinal cord homogenates that have been generated here. The adapter protein ASC is also strongly upregulated. But I told you that there's many different kind of inflammasome. Those are the biosensor that basically sense the different microbes that could be involved. In EAE, we typically induce EAE by injecting, for example, Mycobacterium, which is really important as a trigger for the development of disease. In MS, it's been known that microbes, viruses could be involved also in the trigger of the real disease in humans. So here, what we wanted to know is what could be the inflammasome that could be involved in this response. So we have characterized all of them. This is MEV, which codes basically for pyrin, and there's many of these null-like receptors, type 1, 2, 3, and so on. And as you can see, there's only 4 or 5 of these that are upregulated in the model. We have validated that even more in the reporter mouse line, once again showing that most of these protein involved, most of these inflammasomes are expressed in neutrophils and M1 monocytes. So since there's no good drug that can specifically target these inflammasome cells specifically, I'm not cell specifically, but specifically target the different inflammasome, we once again turn to knockout mouse line. So we studied ASC, which is coded, as I told you, by Picard. And we studied also the 5 node-like receptor that I show you to be upregulated during EAE. But to our surprise, when we look and immunize these animals and look at the clinical score, even though there was some delay in the appearance of the clinical symptoms in this model, in the Picard, MEV, and also NALP1B knockout mouse lines, none of these animals basically recapitulated the effect that we saw in the IL-1 beta knockout. Because as I showed you before, in the IL-1 beta knockout, there's barely any disease. The incidence rate is 30%. Maximal score is about 1 on the scale of 0 to 5. Here, there's a delay of about 2, 3, 4 days sometimes. But whenever they start to be sick, the severity of the disease is identical to the one developed in the wild-type mice. So this really shows us that it seems that the inflammasome are important, but there must be something else that is involved in the cleavage. We think that there could be another enzyme than caspase 1 that could be important in the context of at least autoimmunity. So to narrow down even more the cell population that could be involved What we did is that we used the ASC knockout mouse line that I show you. I told you that there's a slight delay in the appearance of the symptoms, but whenever the symptoms appear, the animal are quite sick. What we've done here is that we wanted to deplete basically the neutrophils and also deplete the macrophages, because those are the two populations that we found are quite important for the production of IL-1 beta. We use an antibody-based approach in that case for neutrophils. It's the anti-1A8 antibody, which has been shown to be able to create a depletion of neutrophils quite effectively. And as you can see, when you deplete neutrophils, there's no change between the ASC knockout and the ASC knockout depleted neutrophils. They look exactly the same. However, when you deplete macrophages and monocytes using clodronate encapsulated into liposomes, it's a toxic drug that you can give to animals to kill these cells, because the phagocyte normally will incorporate that and it will kill the monocytes. Well, you can see that in the ASC knockout, finally, when you deplete monocytes, they don't develop the disease, mimicking basically the response that you're seeing in the IL-1 beta and also in the IL-1 receptor type 1 knockout mouse. So it shows to us that monocyte-derived macrophages somehow are capable of producing the cleaved mature form of IL-1 beta independently of the inflammasomes. This is just to show you that the Picard knockout, which are the ASC knockouts. So it's Picard is the gene name, ASC is the protein name. Well, I mean, they do produce some pro-IL-1 beta. However, if you look at the monocytes from these cells, and if you activate with LPS and ETP, we use LPS in vitro because this is a trigger of the toll-like receptor. So it's the first signal that I showed you before. It's important because it's capable of inducing transcription of the gene that code for pro-IL-1 beta, whereas ATP is the second signal that is important for the cleavage. If you look at monocyte from wild-type mice, in vitro, and you activate with both LPS and ETP, you obviously have formation of the proform and cleavage into the mature bioactive form of IL-1 beta. But if you look at the Picard monocytes, well, you only end up with the proform. They don't produce the cleaved form. It's because, I mean, in that specific case, the inflammasome that, well, the inflammasome are important for the cleavage, and caspase-1 must be important in these conditions. But to our surprise, what we found is that if you take out the Picard knockout monocytes, and if you migrate these cells across an inflamed BBB, an inflamed endothelium, you can actually bypass this response and end up with the cleaved form of IL-1 beta. All that to suggest that we think that crossing the blood spinal cord barrier, crossing the blood brain barrier in inflammatory condition could be the signal that is needed to obtain a cleavage of pro-IL-1 beta into the bioactive form and eventually lead on to inflammation and autoimmunity. At this point, what we are doing, I have a very talented student working on that. This is his ID. His name is Maxime Kuzik. He came to the lab and said, well, I want to understand what are the different enzymes that are important for the cleavage of IL-1 beta in the context of autoimmunity. And he came up with this idea of basically creating a plasmid expressing the pro-IL-1 beta that will be coupled to a biotin ligase turbo ID. And the idea here is that the protein that will be in proximity of pro-IL-1 beta will be captured by this ligase. And eventually you can use that in conjunction with proteomics approach to be able to identify what could be the enzyme that will be important to cleave IL-1 beta, an enzyme that will be other than caspase-1 in the context of CNS autoimmunity. And this is important because what our results are showing is that if you basically block caspase-1, and there's some good drug that can actually target caspase-1, good drugs that can target as well some of the inflammasome, well, it's not going to work in CNS autoimmunity. There's some mechanism that are alternative mechanisms that are involved, and you need to globally understand what are the mechanisms of cleavage in the context of some chronic inflammatory diseases. So what is the effect of IL-1 beta on the endothelium? I told you many times that IL-1 beta is important. Well, if you take out IL-1 beta, the recombinant form, and you activate endothelial cells, you switch expression of many different genes. I told you before that IL-1 beta is a chef d'orchestre inducing transcription of many genes involved in inflammation. If you take out mouse, brain, microvascular endothelial cells and activate them with the recombinant form of IL-1 beta, there's upregulation of GM-CSF, G-CSF, CXCL1, IL-6, CCL2, which is actually the ligand for CCR2. And what you need to understand is that in the context of EAE and MS, all of these factors are key for the development of disease, and that they are downstream of the IL-1 receptor type 1 signaling pathway. They are all important because if individually you take GM-CSF, G-CSF, CCL2, CCR2, CXCL1 and CXCL2, and you knock out these genes, you end up with no disease. So we think that they are important because they are stimulated with IL-1 beta and IL-1 receptor type 1 signaling pathway. And we've been able to recapitulate these effects also using human bone marrow, human brain microvascular endothelial cells also, which is important. To summarize and conclude the first part of my talk, and then I'll briefly go for 5 or 10 minutes with the pain response. What we think is happening is that neutrophils and monocytes that travel in the blood in the context of autoimmunity, they will basically adhere to the endothelium. And eventually they will be recruited in the CNS, passing through the CNS perivascular space. We think that IL-1 beta signaling is really a key signal to open the door to the CNS. We think that without IL-1 beta, it's the key card basically to get in. We think that without IL-1 beta, these cells will not be capable of reaching the cerebrovascular space and eventually doing what they do. Meaning that normally when they reach that space, what they are doing is that they will produce some more of IL-1 beta that will trigger production of the genes that I told you before, many cytokines, many chemokines, cell adhesion molecule, helping to the recruitment of some more immune cells. And also what we found, triggering production of GM-CSF, which is really important for the differentiation of the monocytes into antigen-presenting cells locally in the perivascular space. And we have also shown that this response, production of EPC, is really important for the differentiation of naive T cells into Th1 and Th17 effector cells. So this loop is really important in autoimmunity because those are the cells that are involved in the killing of oligodendrocytes and the generation of the myelin. Now I should move on to the second part of my talk. I told you that when you stain spinal cord and brain tissue with IL-1 receptor type 1 antibody, you see a very intense staining in the blood vessels. It turns out that there are some other cells that also express IL-1 receptor type 1. And some of these cells are actually neurons. You have the great work of Dr. Blakely at your institute targeting some other population of neurons in the brain. But when you look at the spinal cord, there's very few neurons that do express IL-1 receptor type 1. Actually, most of the signal is confined within a population located in the dorsal root ganglia and projecting to lamina 1 and 2 of the spinal cord. And those cells are stained with the orange staining here in the lower panel on your right side. And it nicely colocalized with TRPV1. TRPV1 is it's a channel receptor for the main ingredient in red hot chilies. So it's a channel receptor involved in nociception mainly. And it's also nicely colocalized with P2X3, which is a receptor from some of the nucleotides. Coming back to the DRGs, those are the dorsal root ganglia. This is on your right. This is the spinal cord. I told you that in the DRGs, those are only sensory neurons that are located there. They are involved in nociception. They are mechanoreceptors or proprioreceptors. depending on where they are projecting in the spinal cord. So those neurons are pseudo-unipolar, meaning that they are sending one of their axonal ramification to the periphery, and the other one centrally in the spinal cord. When you stain spinal cord tissue with the anti-IL-1 receptor type 1 antibody, you end up seeing only the terminals located in lamina 1 and 2 of the spinal cord. This is once again nicely colocalized with TRPV1 and P2X3, which are markers, as I told you, of nociceptors. And if you look at the insert here, you can appreciate that down here in the DRGs, this is where are located the cell bodies of these projecting neurons, those that project to lamina 1 and 2. So we did a lot of characterization of these cells in collaboration with Greg Scherer, who's an expert in pain. And we found that this is actually extremely intense IL-1 receptor type 1 labeling in this neuronal population. I told you that 100% of these neurons are colocalized with TRPV1, nearly 100 with P2X3. A lot of them are expressing somatostatin, CGRP, and they are negative for IB4. So we did a lot of characterization, but what you need to remember from this quite busy slide is that TRPV1 is expressed in 100% of these IL-1 receptor neurons in the dorsal root ganglia. Why is it important? Well, just before moving to the next slide, this was confirmed using human tissue that we have obtained, DRG tissue that we have obtained from the NIH. Why is it important? Well, it's important because we wanted to decipher even more the mechanism involved in the pain response. First, we wanted to find out if those neurons are associated with the IL-1 beta producing cells during the disease development. This is the DRGs that you're seeing. I don't know if you still see my cursor. Those are the DRGs. They project to the spinal cord through the dorsal root, and they project into lamina 1 and 2, as I showed you before. If you immunize animal and look at pre-onset, we were quite surprised to see that in the dorsal root, where are the projection of these neuronal population expressing IL-1 receptor type 1, there's a lot of neutrophils and myeloid cells, a lot of myeloid cells expressing IL-1 beta. And if you colocalize the IL-1 beta signal with the LY6G neutrophil marker and IBA1 macrophage marker, they nicely colocalize. Once again, showing that roughly about 70% of these infiltrating neutrophils and 30% of these infiltrating macrophages, well, they do produce IL-1 beta quite a lot in the dorsal root next to the projection of these IL-1 receptor type 1 neurons. So we wanted to assess what will be the effect of activation of these signaling pathways in the development of EAE. So to be able to do that, we initiated a collaboration with Ning Quan, who is the master of generating transgenic tools to be able to study the IL-1 receptor and the IL-1 beta and also alpha system in mouse model. And what Ning has done over the last few years, among many things, is that he was able to generate a global IL-1 receptor knockout, a global mouse line in which all the cells are knocked out in IL-1 receptor type 1. And using this mouse line that he calls IL-1 receptor restore mouse line, you can actually restore expression of IL-1 receptor type 1 cell specifically. So what we've done, because I showed you before that all our neurons are expressing IL-1 receptor, all the neurons expressing IL-1 receptor type 1 also co-express TRPV1. And at the time, there was no IL-1 receptor Cree mouse line. We turned towards acquiring basically a mouse line that was TRPV1 Cree. So by breeding these animals together, you can restore expression specifically in the nociceptors. So you have a global knockout in which you restore expression specifically in the nociceptors only. What happens is that if you inject the recombinant protein into the articulation of the knee, where are projecting this population of neurons from the DRGs, you end up stimulating a pain response that is quite intense. As you can see here in this graph, if you look at the wild type, which is the line in orange, you can see that the pain score, we call that the knee bending pain score in that case from 0 to 5. You can see that the animal will develop early on after 1, 4, and 24 hours, a lot of pain. So if you bend the knee of these animals, they don't like it whenever IL-1 beta is injected. If you look at the knockout, the global knockout, there's no effect. But if you restore expression of IL-1 receptor type 1, specifically in the receptor, you can actually restore the pain response. So this result was really pivotal for us to move on to the model of EAE. Locally, I'm not a pain expert, but luckily, some of my colleagues are. Brad Kerr at the University of Alberta in Canada happens to be an expert in pain in the context of MS and EAE. And what he showed is that the animal that develop EAE develop a pain response that seems to precede the paralysis. And this is really key because whenever you want to investigate the pain response, and this is typically done, for example, using a wound-free filament that you put underneath the paw of the animal, and you see the retraction of the paw of the animal depending on the force of the filament. This is key to have a model where you can actually study this response pre-onset. Because if you go at onset of disease and at the peak of disease, the animal becomes paralyzed. So if the animal is paralyzed, it's impossible basically to study the pain response in this model. So luckily, Brad had shown that pre-onset, prior to the development of disease, of the clinical symptoms, you can actually assess using mechanical allodynia the pain response in these animals. So we did start a collaboration with Brad, also with a colleague of mine at the McGill University. His name is Reza Naini Sharif. He's also an expert. We look at the IL-1 receptor global knockout mouse line. We could not find any fail response to cold temperature, hot temperature, response to histamine, chloroquine. So we did a lot of testing. These animals were normal. However, in the context of inflammation, it was a different story. And to be able to address that, what we've done is that we use the Flux mice that has been created by Dr. Blakely. So I'm lucky because today I'm speaking in front of the two main creator of these mouse models that we've used in our work. So the IL-1 receptor Flux mice. And in that case, what you can do is that if you breed with the TRPV1 Cree, for example, you can generate a conditional knockout that will be cell-specific, in that case, the receptors. This model was important for us because we figure out that if we wanted to study the disease, the receptor, IL-1 receptor type 1, needs to be in the other cells. Because I showed you before, if you study the IL-1 receptor type 1 global knockout, there's no disease. So you won't be able to study the pain response in the absence of disease. So it was key because we wanted to target the deletion specific in the receptors, but we wanted to keep expression in endothelial cells and immune cells as well. First step is that we once again injected IL-1 beta into the articulation of these animals. We found that if you do that, and if you look at a marker of neuronal activation, IL-1 beta will trigger activation of the production of phosphoprotein, which is a marker of neuronal activation, specifically in the TRPV1 and also IL-1 receptor co-expressing cells. However, if you look at the IL-1 receptor global knockout, this response is gone. Not surprising. You get rid of the receptor, you inject the cytokine, you don't have a response anymore. It's logical. And when you look at the CKO mouse line, which was the most important result here, is that we were able to completely block activation of these neuronal receptors. So that really brought us, and this is almost one of the last slides of my talk, that brought us to the model of EAE. First of all, I showed you before, if you immunize animals, they will be sick. They show clinical symptoms that will be, clinical score will be between 2 and 4 typically. This is the wild type animals that you're seeing here. I show you as well, and this is shown once again here. If you look at the knockout, global knockout of IL-1 receptor type 1, there's basically no disease. But if you actually do a CKO specifically in the receptors, the disease will develop normally. And this is kind of what we were hoping in that case. I'm not going to lie. I mean, if it would have been, the disease would have been affected, it would have been very difficult to do the experiment, but it is what it is. And we were lucky enough that if you get rid of the receptor and the receptors, there's no effect on disease development, no effect on inflammation. Compare this one here. This is Cd45 staining that shows infiltration of leukocytes in the spinal cord. If you look at the CKO mouse line, there's a lot of inflammation, and this fits quite nicely with the clinical score. However, panel G is probably the most important here. If you look at the pain response using the wound-free filament, if you do and put some pressure underneath the paw of the animal, what you will see over time with the orange line here with the wild type is that more the disease will develop, the more the animal will become sensitive to pain, and the less the force of the filament will be needed to create the pain response and retraction of the paw of the animal. If you look at the global knockout in blue, the response is completely gone. And when you look at the CKO mouse line receptor, the most important results of this part of the talk is that you can get rid of the pain response. So it really shows that IL-1 receptor type 1 in nociceptors is not important for disease development and inflammation per se, but has a key role in the nociceptive response. To summarize, myeloid cells, we've shown that myeloid cells produce IL-1 beta during inflammation-associated EAE, mainly neutrophils and monocyte-derived macrophages. And I've also shown you that the monocyte-derived macrophage seem to be capable of producing IL-1 beta using an alternative pathways that does not involve inflammasomes and the typical caspase-1 enzyme. I've also shown you that CNS endothelial cells express IL-1 receptor type 1, and that there's also a subset of nociceptors that are capable of heavily expressing IL-1 receptor type 1. Finally, but not least, I've shown you that IL-1 receptor expression in CNS endothelial cells is really key for the development of EAE disease, while the IL-1 receptor type 1 expression in nociceptors appears to regulate pain in the context of chronic inflammation at least. So on that note, quit the presenter mode. On that note, of course, I would like to acknowledge the number of collaborators that I have. Ning, I mean, you're not on that slide, but you're probably the most important one. My team, of course, that has done all the work. So that's been truly a blessing to be able to collaborate with all these great people. And on that note, and I'm sorry, because we are probably 5 to 10 minutes late because of the slow start that was due to the problems that we had with the Zoom presentation. But on that note, I'll take any question, and I'm going to leave the sharing mode to be able to hopefully see some of the question in the Q&A.
Thank you, Steve. That was a wonderful talk. And Randy apologized that he had to go to a doctor's appointment at the end of your talk. So I don't see many more questions, but I have some questions. Do you have any ideas in terms of this non-inflammasome induced mediated activation? I think you are getting on a terrific project on that, because I know, like in the brain, maybe some of the expression of the IL-1 may indeed independent of inflammasomes.
Yes. Well, I mean, we're doing this experiment at the moment where we use the Turbo ID method to be able to identify some of these interactors. To be honest, we don't have any list of protein that they could give you at this point. There's some enzymes that have been shown over the years to be at least capable in vitro to cleave IL-1 beta, as you know. Many of these enzymes are produced by neutrophils, for example. I can think about elastase that has been shown before to be capable of cleaving it, and some metalloproteinase. Right from the start of this project, we took a different approach where we were trying to inhibit all the caspase, all the metalloproteinase, and also some of these the elastase, using some drugs that will inject to the SC knockout animal in vivo. But we really were unable to find and identify a single protein that appears to be key for the cleavage. And so that's why we decided to choose a different approach. Obviously, with the injection of drugs, you're always limited for the access to the CNS because of the blood-brain and blood-spinal cord barrier. So it's possible that the drugs that we have injected IP mainly or subcutaneously were not able to reach their goal, which was the CNS. So I cannot, claim with certainty that those enzymes are not important, but they don't seem to be part of these categories of enzymes.
I have a question from Larry.
Can you hear something, Ning?
I'm looking at Q&A. There's a question from Larry Toe. He's asking, have you looked at any other pain inducers other than the EAE and administration of IL-1 itself?
The answer is no. As I pointed out before, I'm not a pain expert, and this is not it was not the main focus of the lab when we really started this work. And it happened by luck that we detected expression of IL-1 receptor in these receptors. And we became interested through collaboration as well with some good colleagues such as Brad Kerr and Reza Naini. And luckily, because of them, I was able to do these experiments right from the start. Obviously, over the years, we have learned how to do these experiments ourselves. But the short answer is no. We have not tested any other blocking agents or drugs in the context of the pain response that develops in EAE.
Okay. Here's another question from Lillian, and she's wondering, have you looked into the special MMP9 produced internally by neutrophils, constitutively or maybe even induced?
Yeah. The answer is yes and no. MMP9 and MMP2 were two of the main targets that we're after at the beginning of this identification of the alternative pathways of cleavage. We've done some injection of some drugs that can actually target. I don't know if there are specific drugs, as I pointed out. I don't recall the names. They were quite complicated names, but if I recall, they were targeting both MMP2 and MMP9. And we failed to demonstrate that those are the enzymes important in the cleavage. But as I said before, it could also be because our drug were not capable of crossing the blood-spinal cord barrier in our case. So I have not discarded completely MMP9. We discussed at some point in the lab the possibility of having the double knockouts, but we have not done that yet.
Okay.
It's still on the list. It could be a very good candidate. It's still on the list.
Yeah, Lillian herself has done some really good job of work on the MMP9 actually may even be involved in the EAE disease itself more than just pain. So that's why I'm asking that kind of question.
I'm gonna note the name and I'll go right away, right after, to be sure that I read this paper carefully. What I should say at this point, if there's anyone that has any question for me, if you want to reach out, if you want to talk to me over the next few days, anytime. So just drop an email, send me an email, and we can have a good discussion about all of that.
Have you,
Awesome.
do you think because typically IL-1 could stimulate the endothelial cells to produce COX-2, which might also be involved in pain. Is there any sign in that at all when you give IL-1 to the knee?
Yeah. When we give IL-1 to the knee, I've never looked at the expression of COX-2 into the blood vessels. Obviously, I know from the previous work that we have done and that you have done also, Ning, that if you administer IL-1 beta IV or IP, you have massive expression of COX-2 throughout the blood vessels. The short answer is that when we inject in the articulation, we've not looked at this. I would not be surprised if when you do the injection in the articulation, it will eventually reach at least some part of the blood vessels and eventually end up stimulating COX-2 throughout a major part of the CNS. So I think to be able to really address your question, we will need to go with extremely fine amounts of IL-1 beta to make sure that it remains local. And then to be able to study if COX-2 could be even expressed in the neurons itself. Endothelial cells perhaps, but maybe in the neuronal population. It's not impossible.
Any more questions from the audience? Well, if not, I would like to give my thanks to Steve again. It's for such a wonderful talk, and I'm sure there are students that and postdoc might contact you after this to because some of us are working in a similar area, so hopefully we can have more fruitful collaboration and interactions.
I would love to. If anyone wants to contact me, as I said, feel free to do it. Don't be shy to do it. And again, thank you so much, Ning, for this very nice invitation.
Yeah, of course. Well, we may do it again.
We will do it again, for sure.
All right.
Thanks, everybody.
All right. Bye.
Take care.